Great. Thank you very much. And thank you for inviting me to speak today and elucidate some more on the poster that I won the prize with in Montreal. So today I want to talk to you about ASO activity, comparing it on protein versus RNA level, basically. And as said, yeah, I'm a postdoc here in AstraZeneca in Gothenburg at the Safety Sciences Department. And yeah, my focus is on oligo safety in that regard. So if we boil it down to the basics, what determines ASO gapmer activity and toxicity, we can have three parameters, basically. We can have the sequence, we can have the design, and the chemistry. I'm no chemist, so today I'm not going to talk too much about design and chemistry, but what intrigues me in this is that antisense oligos with a slightly different sequence can have such a vastly different activity and toxicity profile. And some of that we know it can be explained, for example, by off-targets or protein interaction, but I think this whole puzzle has not yet been solved, and that's what I'm working on in my postdoc project and what motivates me every day to go to the lab. So for this particular study that I want to talk about, it's about ASO activity on RNA versus protein level. So currently in the field, what we are doing, looking globally at ASO activity, there's a strong focus on the RNA level, for example, with transcriptomic, or as it's also called, RNase H, even though most of the therapeutic effect, we would observe it on the protein level. We think this could be due to previous limitations that we are not studying the protein level on a global level. Could be cost, depth, throughput, but also protein half-life can play a role, because yeah, if the protein that we are studying has a long half-life, there might be a delay in the response on the protein reduction. We believe here that for this study, we solved most of these previous limitations that will then allow us to study ASO activity on RNA versus protein level. So we got access to the internal, what we call fast proteomic platform. It's giving a depth of up to 6,000 protein per sample in a 96 well plate, and it could be run on the throughput of 60 samples per day, which allows us to also then sample for these different time points to account for the potential impact of varying protein half-lives. And I should say, of course, as well, that for the RNA-seq, we also had access to a high throughput platform that allow us to run this study that I'm going to describe next. So to boil it down into two questions for ease, the study aim, we first asked, can we use these RNA-seq and proteomic high throughput methods to distinguish safe from toxic ASOs? So to do that, we identified two compounds from the literature, characterized their toxicity, optimized conditions to run the proteomic, and then did bioinformatic analysis that I will tell you about to see if we can really distinguish them also using the omics methods. And for the second question, we are really interested on the ASO activity. How does it compare on RNA versus protein level over time as well? And for that, we are really looking at on-target activity, but also the unintended off-targets. So yeah, let me jump into the first question then. Identification of the tool ASO. So we found 5 ASO compounds in the literature. They have a similar design and chemistry, so they mainly differ in sequence. And we can see that already that gives a different safety profile or toxicity profile, if you will. 1 and 2 are very safe based on literature finding, but 3, 4, and 5 would be considered toxic, both in vitro and in vivo. So what we first wanted to do is confirm these findings in our own acute tolerability study. So what we did there is we inject at a high dose subcutaneously these ASO gapmers, and then after 3 days we collect serum ALT, which is a marker for liver toxicity, and we also performed histopathology on these mice. But for today and for simplicity, I will only show you the ALT results, even though they correlate with the histopath. So we can see that the PBS control is at the same level as ASO 1 and 2, which were considered to be safe. We can have a medium toxic ALT activation for ASO 3, and 4 and 5 are really super toxic. They are like, I would say, off the chart toxic, because 10,000 is the maximum of the assay that was run here, and these samples were already diluted. So yeah, really toxic ASOs, these 4 and 5. So next, we wanted to find an in vitro system in which we can do the omics methods that correlates with this data. And of course, we know from previous publication that HepG2 liver-derived cell lines could be a good tool for that. So we did a screen there for the different time points to see where is the peak of the caspase activity, and is this something we want to use as well. So we realize the caspase 3,7 activity, which we use as a proxy for apoptosis, it peaks at 48 hours in this particular experiment upon transfection. And then at, for example, 39 nanomole here, we can nicely see a separation again between the different ASO compounds. So there is a safe one, a medium toxic one, and 3 toxic ones. And if we take this concentration in which we get the nice separation and then look at the time course, we can also appreciate again, the peak at 48 hours, and that it's quite persistent here in the assay, the caspase activation, and that ASO1, for example, is very clean, and ASO2 also cleaner than the rest. But unfortunately, we couldn't really use this condition to, of course, run the proteomic, because this is 384 well plate, and we need a bit more sample for the proteomics. So we try to see if we can recapitulate these results now upon optimization. So increase the starting cell number from a few thousand to 100,000 cells in the 96 well plate. And we do the same thing again, except this time we are also including cell titer, which is a cell viability marker, because we want to be sure that we are not killing the cells in this particular assay, because then it will be hard to do proteomic or RNA-seq on it and get meaningful results. I should mention here now we also took the 50 nanomole ASO concentration only to focus on. And then what we can see is that we get similar peaks in the ASO caspase activation, again, in the FG2 cells with these higher cell numbers. So the toxic ones are showing the highest activation around 48, 32 hours, as we saw previously in the 384 well plate assay. And most importantly, we get a good cell viability. So for most of the sample and most of the time, it's above 80%. So we will not have dead cells or cell debris that could potentially ruin the proteomic or RNA-seq assay here. So with that, we were very happy. We proceeded with the study, and I want to describe the design here in the next slide. So yeah, as I was saying, 50 nanomole transfection in HepG2 cells. We take RNA-seq at 24 hours because we know in this in vitro transient transfection system, we get already after 24 hours a good knockdown or maximum knockdown activity. And for the proteomics, we are sampling here 6 different time points to account for this potential impact of the varying protein half-life that I mentioned before. And then one of the bioinformatic analysis we did is just look at differentially expressed genes or differentially expressed proteins with the parameters that I'm giving here. And already there, I think we can appreciate the similar picture as the caspase and the ALT results in the mouse, that some of these really toxic ASOs show a lot of changes happening, both on the RNA level in blue, but also on the protein level in light green. And yeah, the one that we considered medium toxic in those assays, they show lower number. And one seems really quite safe here because almost nothing is changing here on the global level. Now, another type of analysis that we can do here to see if we can distinguish safe from toxic ASOs would be the principal component analysis. So here on the left, I will start with the RNA-seq. I think we get a quite nice separation here because we have the mock control here that just has the transfection reagent, and that's forming a group together with the safe ASO1 that we saw before is safe in most of the assays that we have run. And then we separate a nice group of ASO2 and 3 that we consider here to be medium toxic based on the PC2 component. And the highly toxic ones in the mouse and in the HepG2 caspase assay, they separate both on PC1 and PC2. So on the RNA-seq level, we can use PCA to distinguish safe from toxic sequences. But can we do the same for the proteomics, you may ask? Yes. We can do it for most of the time points from 32 to 72 hours. But here I'm showing as an illustration the 48-hour time point. And we can appreciate hopefully the same picture again where mock control is clustering together with the safe ASO. We separate based on PC1 and 2 medium toxic ASO compounds, and then the really toxic ones, we separate them from the controls based on the PC2. So that is, in our opinion, a nice success for the first question, basically. So we pick 2 compounds from the literature, characterize them in vivo, in vitro, and then we use this RNA-seq and proteomic method to get a similar classification in our assays. So then I want to move on to the second question, which is about the activity part, the ASO activity. So first we can have a look at the ASO on-target activity. So in our cell model for ASO3, we have an on-target there that we can reduce. So as I said, the RNA-seq was only done at the 24-hour time point. So we get a nice downregulation there. And surprisingly, what we see for the proteomics as well is that maybe there's no impact of this prolonged half-life for this particular on-target protein, because already at 24-hour, we are very significantly downregulating it. And on top of that, it's already going back to mock levels, this protein, after 56 or 72 hours. So that was a bit puzzling to us. It is counterintuitive to our hypothesis, because our hypothesis was that the protein half-life might delay the proteomic response to the ASO on target as well. So we wanted to validate that basically orthogonally. And to do that, we used the Western blot and the qPCR. qPCR to measure the RNA levels of the on target, and the Western blot for the protein levels. And then what we can appreciate now, we get also the time course added for the RNA level. So here, again, on the RNA-seq, we only have the 24-hour time point. What we see on the qPCR is that the silencing of the target RNA already happens 8-hour onward. But also there, we see that it's coming back to control levels after 56 and 72 hours. And I hope you can appreciate as well in the Western blot picture, A is the ASO treatment, M is the mock, and depicted with the stars here that early on, there is a big difference between the mock and the ASO, but it evens out again towards the end of the assay. So against our hypothesis, there is actually an overlap of the time course for RNA and protein level. To really get a better idea of that, we wanted to go for other cell models as well. So we usually have good results in long-derived cell lines as well. So we took the A549 and the PC9 cancer cell lines, and also looked at the ASO3 on target there by Western blot and qPCR. And we see a similar picture there. So if we look at the RNA level here by the qPCR, we see early on already it's downregulated nicely by the ASO3, and then by the end of the assay, it's coming back up to control levels. And this is also confirmed then by the Western blot here, where we see early on a lot of loss of protein, but then towards the end of the assay, we are getting back to mock levels. And we can see a similar picture for the PC9, although there is a different degree of silencing activity between the two cell lines. But yeah, the trend is basically going down in the 24 hours, coming back towards the end of the assay. And again, here we confirm it by Western blot as well. So that gives us more confidence in those results. And on top of that, you might also think maybe it is an effect that is specific to this particular ASO3. So we wanted to test 2 more ASOs as well. And what I should note here is then in this long-derived cell line, transfecting the same amount of ASO, we do get, interestingly, a higher knockdown efficiency actually than in the HepG2. But then to look at 2 more additional ASOs, we just do it by qPCR and Western blot because we didn't have them in our omics data set. Again, I would argue we can see a similar picture. So on target 2 with the additional ASO 2, we can see, again, peak degradation, 24-hour, coming back to mock levels towards the end of the assay. And this is well reflected in the Western blot again. And for the last ASO, again, you're not going to be surprised by this probably, but again, we see a similar kinetic here in this HepG2 cell line, like, although again, different degree of knockdown, it happens early on, goes back to mock levels, and we reflect it again in the on-target Western blot. So I think with that, we nicely characterize the on-target kinetics. But what is also really interesting to us, and what sparked a lot of discussion at the actual OTS meeting in Montreal when I presented my poster, is of course the off-targets, because that is a safety concern that we are currently finding more and more details out about, and it's important to address. So let me go into the off-target then. So for off-target, we can predict them based on sequence homology, but that's not the only thing we did here. We also focus now on the overlapping data set of the RNA-seq and the proteomic, so that we are looking at off-targets that are expressed on the RNA and protein level. So between the 5 different ASOs, we have different numbers of predicted and detected in our data set. And then we can start looking, are they down on the RNA level? Are they down on the protein level? And are they down on both? Or are we missing something by using either method? So on the RNA level, we can see a lot of downregulation. If we take again ASO3 as an example, where we have 240 predicted and detected off targets, more than half of them are significantly down using our parameters on the RNA level. But what was surprising to us is that actually on the protein level, there seems to be less of a downregulation or number of downregulation using our parameter. It's almost for ASO3 10 times less. But I think what is the most important one here is we look at the overlap. So if we find something down on the protein level that is a predicted and detected off target, do we also catch it by the RNA-seq that we are currently using to assess these off targets? And for the majority of them, we actually do. So it's not like we are missing out in this experiment any potential important off target hits that are down on the protein level by using RNA-seq. One other thing that we wanted to look into is the magnitude of the changes. So how much is the RNA versus the protein level reduced? And for this, we took a, I would say, simple formula where we take the RNA or RNA-seq log2 fold change and subtract it with the maximum protein log2 fold change. And then what happens in all these graphs for the 4 different ASOs that have predicted and detected off targets, we see mainly blue in the graphs and a shift to the left, which means that the change is always going to be bigger on the RNA level here. So that could maybe give an indication also to the different numbers that we observe on downregulated on protein and RNA level. Then for the last pieces of data, I want to also validate some of these off targets here again for ASO3. So this is one of the off target that we could find for ASO3, forms a very similar pattern compared to the on target. So there's downregulation peak for proteomic 24-32 hours, and then it goes slightly up again, but it's not as dramatic increase or going back to control levels as for the on target. And I should also note that clearly the level of the silencing of the off target, because there's 2 mismatches to the off-target transcript, is lower than for the on-target. So the on-target silencing is more efficient. So again, here we are going to confirm the results using the orthogonal validation of the Western blot and the qPCR in these HepG2 cells. And we could again basically confirm that the kinetic also for these off-target, as I showed you extensively for the on-target in HepG2 cells in different cell lines, it seems to overlap. So with that, I just want to come to the conclusions already and basically walk you through what I've shown you today. So first, I explained the experimental approach that we could take and overcome some limitation to compare global RNA versus protein levels. What we saw to answer the question 1 is that we can use both methods together with the principal component analysis to separate safe from toxic tool ASOs. What was also remarkable is that we see a higher number of ASO off targets with reduced RNA level compared to protein level. Again, remember for ASO3, there was almost a 10-fold difference. And also the magnitude of the off target changes or the off target reduction are greater on the RNA level than the protein level. And then finally, of course, against our hypothesis of the delayed protein impact, we actually do not see this. We do see a similar time course or almost overlapping time course for ASO activity on both the on and on target off and on target levels. And to conclude with that, I think that we can say that global proteomics could serve as an important complement to our current RNA-focused ASO activity and off-target assessment. But of course, more studies need to be done to see how we can actually leverage it to the best potential. And with that, I am coming to the acknowledgement. I really want to, of course, thank OTS again for inviting me here to speak in this webinar, but also a big thanks to my manager here at AstraZeneca, Patrick Anderson, and a very close collaborative team. So we have people here from 4 of the AstraZeneca research sites that have been important in helping to realize this project and this study. So thank you very much for that. Thank you for your attention. And I would be very happy to answer any questions you might have.
Yes, thanks a lot, Daniel, for this great presentation. I think very puzzling results sometimes. So maybe to start with the very first question. So because you mentioned ASO 1, 2, 3, 4, 5, and you based that on literature. So can you comment on the targets? Because I can assume that these are known from literature.
Yeah, I think it's one of the caveat of we are focusing here a lot on safety, I would say. So again, I show a lot of on and off target for ASO3. So in this particular human cell line, that's the only of our 5 that has a target in there, basically, if that answers your question.
Yeah, but what is the target? Can you specify the target as well?
Well, I guess I'm allowed to say this PCSK9.
Okay. with all of them?
No, what I'm saying is, so we base the whole idea of picking the two ASOs based on the safety profile and not that they will have a target in the human cell lines that we are using. So I'm saying that only ASO 3 out of these 5 ASOs has a human target has a human target.
Yeah, because it's exactly where I want to go. Because you do your RNA-seq analysis. And I kind of assume that, especially for ASO3, there might also be an on-target effect, right? So you have downregulation of your target of interest, and also that might have an effect on all kind of downstream pathways. So how can you make the division between the on-target effects and the off-target effects? Because that might be quite difficult to.
Yeah, I think I agree with you. I don't know if I have the perfect answer to that, but maybe shortly I might say in my study I cannot make exactly this distinction, but I would highly recommend to read Patrick and other industry people latest paper in the, I think it's the OTS journal, with the recommendations for the off-target assessment. And basically, I think if I recall correctly, or that might have been internal discussion, the idea is to choose the 24-hour time point and not the highest concentration, is to make sure that these potential on-target toxicity effects are not yet occurring. So we are really looking at the primary off-target effects by choosing not the highest dose and an early time point, because we expect like secondary off-target effects not related to the sequence to occur later on. So that's why we pick always the 24-hour time point for these off-targets.
Oh, good. Thanks. Yeah, that kind of makes sense. I also have, so can you comment a little bit on the delivery method? How do you deliver the cells?
It's like normal liposuction, I would say, is lipofectamine 2000. Yeah, I think like many people have used in the field, so I don't think there's much special about it, except for, yeah, of course, we would want to do maybe gymnosis as well, but for this particular study, we didn't choose to do it.
Yeah, because I think what you showed during your presentation is that you already had your very first effect on RNA after 8 hours, but you had a similar effect already also on the protein level.
Yeah.
So can you maybe, especially for target number 3,
Yeah.
do you have any idea what the protein half-life is of your target?
It's again a very good question, things that we are constantly discussing. I have a rough indication based on data from other cell line, but in this particular cell line, I don't know it. But for the on target, if that's what you ask, it's supposed to be around 44, 48 hours of protein half-life.
Okay, because then one might not expect too much on target effect already after 8 hours, right? So that
No,
might be
that's
a
true.
little bit
That's why we
puzzling.
were a bit puzzled by this, yeah. But you think it
Any
might
hints
have
for
to do
that?
with the transfection method? Is that what you're saying here?
It can be, yeah. Not sure. Let's quickly go to the Q&A. So if you have a question for Daniel, please feel free to use the Q&A function. So let's go to the first question. Question from Noah. I think that might be something. I will just read it. How do you distinguish between true off-target knockdown and exaggerated pharmacology? Knockdown of intended target causes reduction in expression of another gene. Yeah, I think this is already a question that we asked before. So maybe I'm not sure if you have to add something.
No, I think yeah, except maybe to elaborate again a bit more on how we predict the off targets, and that's based on like a basic mismatch criteria, allowing up to 2 mismatches, but also insertions and deletions.
Yep. Question from Sanjay. How many differentially expressed genes were actually protein coding? I think you showed it on slide 13 or 14. Do you have an estimation for that?
I think I don't have it on the top of my head, sorry, no. But it's a relevant question. I will definitely have a look at that, yeah.
Yeah, that might also be quite relevant for your studies.
Yeah, no, good point.
Quick question from Sabina. In your proteomics approach, you use transfection to get the ASO into the cell. Did you consider to test gymnotic conditions as well? where you may get a different protein binding profile. Yeah, also something that we discussed already before. So maybe something to add to this or to comment on this?
Yeah, I don't want to comment too much on ongoing studies, but we are clearly considering it and probably going to run, I think, also like gymnosis for this. But then it would not be in the HepG2 cell line, I would have to say, because at least in my hands, I don't get too much of a toxic response in there upon the gymnosis. But I think I think Sabine has published papers or been part of papers, or her company has been, where they are using nicely gymnosis in primary hepatocytes, for example, and we get a nice toxicity response. So that could be something to consider.
No. This is true. Yeah, I was thinking. So I think I'm not sure whether it's super relevant, but you during your studies you had a lot of ASOs, all gapmers. Are you also considering to take along other kind of ASOs? So, for example, splice modulating ASOs.
Yeah, I think it is a relevant question again, but let me think one sec before I answer. So yeah, I would have loved to include something like that, is basically going to be my answer. But what I'm struggling with, like going to conferences like OTS, talking to SSO people, is finding as toxic sequences as this in terms of splice switcher. Probably again, if they are toxic, the toxicity mechanism might be different. I'm not an expert in there, but of course I would have loved to work with some of them if I can get like similar toxicity responses from them in, for example, the caspase and things like that. But I haven't managed to find compounds like that. Otherwise, I might have included them.
Yeah, and based on your proteomics or your RNA-seq experiments, so could you identify a few more pathways, that are common in the ASO 4 and 5 or 3, 4, and 5
Yeah,
that you
yeah.
do not see in ASO 1 and 2? So that might be good
No, thank you for this one. I
explanations.
think you are really asking the relevant questions here. So we tried to do like pathway analysis or gene set enrichment analysis, if I say it correctly, on all different type of combinations here, and also in a more unbiased way. And we never really find, unfortunately, anything that is like common between the toxic ASO. It's always like specific for each toxic ASO by itself. But I think also that is what others have found previously in literature. I think doing transcriptomic studies on mice dosed with toxic ASO, but also on cell line, if I recall correctly. So yeah, of course, if we would find that, that would be amazing. But I think it's safe to assume that nobody's, or we might not find that.
And maybe not pathways, but then just particular genes or proteins that you can identify?
No, unfortunately not, but I'm still digging a bit more in the data. For example, for the PCA, like some principal component, you could try to dive in deeper, see what is the underlying variation there, and see if there is interesting genes in there. But in my mind, also maybe with a bigger ASO subset, that would be also more appropriate, and maybe we could start to see some more similarities there. But we are not there yet.
No. Question from Jean-Christophe. Can you somehow predict those RNA of targets that do not translate into protein reduction, or the other way around? If yes, based on which criteria?
Oh, I don't think we can predict that. Maybe that's going to be sort of the ultimate model, right? If we are going to run a lot of these studies and find out the results, and then we could feed it to a model. I don't want to say AI or machine learning too much, but I mean, after many iterations or with a bigger data set, maybe we could come up with some parameters like that and start refining them, basically. But I think based on this, it's still hard to comment anything on that or even predict. No, that will be difficult.
Yes, and then we have a question from Wenwu. Based on your predicted off-target and RNA-seq off-target data, are there patterns or mismatches that you see that causes an RNA silencing where others would not? For example, double mismatches linked to each other, or double mismatches 10 nucleotides apart, or single mismatches in the middle versus at the beginning or the end. So could you do any predictions based on that, on the sequence homology?
I think we tried to look into that, but I'm not sure. I'm not too much on the bioinformatics side myself, but I think we might have also too low numbers to predict that. But yeah, Again, I'm not the one making the prediction, so.
Okay, yeah, I was a bit surprised by the number of samples that you could do in a day. I think you could do like, I'm not sure, what did you say, 60 samples you could
Yeah,
analyze per day.
yeah,
So that's at least for an academic, that's quite a huge
yeah,
amount of
no.
samples.
But that's something that the guys here in the US they pioneered that, and actually I think they improved the technology even further than that. So.
Okay, let's say I have a target and I developed an gapmer ASO, and that seems to be working pretty well both in vitro and in vivo. And then I need to go to the whole tox screening platform. So what would you advise me to do as an academic with not that much money, but what kind of experiments would you do first?
Ooh, that's a tricky question. I think again, as the literature has shown, and as I showed here, I think the caspase could be a very good option, and I think affordable as well. Uh, I'm not too much in. I'm not sure about the market prices of the RNA-seq, but I think it would be a good start there as well. And I know there is, for example, CRO companies that would also, do all of that. But yeah, I think RNA-seq, caspase, they would be a good indicator to predict, on the off-target, but also on the toxicity, I would say.
A follow-up question from Jean-Christophe. So besides the mismatch counts and positions, do you also use the delta G to predict the RNA off targets? Or do you have a preferred method?
Ooh. Again, since I'm not the bioinformatician that worked on this, I think I have to refrain from giving the answer here, because otherwise I'm risking to give the wrong answer, I think, and that would also not be nice.
Okay, cool, thanks. Another question from Noah for the tox. So are you aware of any organ on a chip assays where you can do these tox assays?
Ooh, yes, but not that much recently. I know we had a postdoc working at Astra, for example, on the kidney on a chip, where they did some toxicity assays with LDH, for example. But currently, there's nothing that jumps to mind immediately that I could mention. Sorry.
Cool. Well, thanks a lot. So I give the word back to Aaron, I think. I don't give the word back to Aaron. Thank you everyone for joining today. Thank you, Danielle, for presenting your work, and see you all next time at the ODS webinar series. So goodbye.